Usually when we talk about a blueprint, there is this expectation of like rigid, unbreakable consistency.
Right. Totally.
You build a house, and the living room and the kitchen both originate from the exact same architectural drawing. The lines on the paper dictate the walls, the windows, the doors. It is completely uniform.
Which works beautifully for concrete and wood. It is a single master plan that governs the entire structure, and nothing really deviates from those drawn lines.
Exactly. But then you step into the human body, and suddenly that entire blueprint metaphor just breaks down completely.
It shatters, honestly.
Yeah, because every single cell in your body, your brain cells, your heart cells, your liver cells, they all contain the exact same master plan, the exact same DNA, yet a brain cell somehow knows to be a brain cell. It doesn't, accidentally start beating like a heart muscle, even though it has the instructions for a heart muscle sitting right there in its nucleus.
It is the absolute definition of a biological mystery. If the genetic code is identical across all these incredibly diverse tissues, where is the instruction manual that tells the cell what to actually do with that code?
Right. Yeah.
Like, how does a cell know which pages to read and which pages to just, staple shut?
And that question is exactly what brings us to today's deep dive. We are looking at a stack of sources, primarily a really groundbreaking 2026 Nature Methods paper that introduces a software package called CREsted.
Yes, CREsted.
Yeah.
Which stands for Cis-Regulatory Element Sequence Training, Explanation, and Design.
Right. Quite a mouthful. But today, our mission for you listening is to explore how artificial intelligence is not just reading the hidden instruction manuals of our DNA across different species, but how it is actually starting to write brand new synthetic DNA from scratch.
Which is huge. I mean, we are moving from observing biology to actively programming it. And to understand how profound that is, we really have to look at the hidden manuals themselves. Biologists call them enhancers.
Or cis-regulatory elements, right?
Exactly.
Okay, well, let's unpack this a bit. Before we can understand how an AI writes DNA, we need to understand what it is actually reading. What exactly is an enhancer? Because, we all learn about genes in high school biology, but enhancers usually get completely left out of the textbook.
They really do. Think of enhancers as the genetic control panels that drive cell identity. They are sequences of DNA that don't actually code for proteins themselves.
Okay, so they aren't the building blocks.
Right, they aren't the bricks. Instead, they control when, where, and how strongly a nearby gene is turned on. They function by integrating specific combinations of what we call transcription factor binding sites.
Transcription factor binding sites? Break that down for me.
Sure. So transcription factors are specialized proteins that float around in the nucleus, and they bind to these specific enhancer sites on the DNA to either activate or repress a gene.
So it is essentially a physical docking station. A protein lands on the DNA, and the gene turns on.
Yes, physical contact is required.
But it can't just be a simple on and off switch, right? Otherwise, we wouldn't have thousands of different highly specialized cell types in the body.
Oh, it's far from a simple switch. The logic of these enhancers is inherently what biologists call degenerate.
Wait, degenerate? That sounds like, a biological insult. What does that actually mean in this context?
No, it sounds bad. But in biology, degenerate logic just means there are many different paths to the exact same destination.
Oh, okay.
Yeah, a single transcription factor can actually recognize and bind to many slightly different DNA sequences. And completely different combinations of these binding sites can drive the exact same functional output in the cell.
Okay, I see. It is like a complex mixing board in a recording studio.
That is a great way to think about it.
Yeah, like you don't just push one single button to get a hit song. A single transcription factor isn't just an on switch. It is the interplay of their strength, how many copies there are, and their specific arrangement on the DNA that creates the final sound, or, the final cell identity in this case.
Exactly. And because it is degenerate, you could theoretically achieve the exact same song using a slightly different combination of dials and sliders on the mixing board.
You're spot on. But let's take that mixing board a step further. In this biological studio, the dials are constantly moving on their own, depending on the environment.
Right, the cell is reacting to things.
And the board itself is unimaginably massive. Human researchers have struggled for decades to decode it manually because the patterns are just way too complex for the human brain to parse.
Totally. And that is exactly why the researchers built CREsted. They needed a deep neural network to decipher this incredibly dense sequence grammar.
But, I mean, how does the AI even look at the mixing board? You can't just feed it a giant soup of genetic material and say, hey, figure it out.
No, you definitely can't. So they structured CREsted into 4 distinct modules. First, they start by finding the active parts of the board using a technique called single-cell ATAC-seq.
ATAC-seq, what does that do?
It measures chromatin accessibility. You see, DNA is usually tightly spooled up in the nucleus, wrapped around proteins like thread on a spool.
Right, just to fit inside the cell.
Exactly. So to be read, the DNA has to physically open up. ATAC-seq basically finds the open parts of the DNA in a given cell.
So it is isolating which specific pages of the instruction manual are currently dog-eared and being read by that specific cell.
Precisely. That is the pre-processing module. Once they have that open data, they move to module 2, model training. They train sequence-based deep learning models to predict that accessibility directly from the raw DNA sequence.
So the AI looks at the raw sequence letter by letter, A, C, T, G.
Yes. And then in module 3, interpretation, it calculates exactly which short sequences or motifs are driving a cell's identity. It gives these sequences nucleotide resolution contribution scores.
And then there's a 4th module, right?
Right, the design module, where it actually builds synthetic enhancers using in silico evolution. But we will get into that wild part later.
Let's do a quick recap for everyone listening. Point 1, enhancers drive cell identity using a complex degenerate code, basically a massive mixing board. Point 2, CREsted is a 4-step AI tool designed to decode and eventually design these enhancers.
That sums it up perfectly.
So if you are listening, think about that mixing board analogy. If you could isolate the specific audio track for any cell in your body, which one would you actually want to listen to? It is a wild thought.
It really is.
But moving forward, if this AI is learning how to read the dials, what does it actually do with that information? Like, the researchers didn't test this on a simple single-cell organism, did they?
Oh, not at all. They jumped straight to one of the most complex structures in the known universe, the mammalian brain.
Wow. Straight to the brain.
Specifically, the mouse motor cortex. They built a specialized model within CREsted called DeepBICCN2. And to train it effectively, they used a really clever 2-step procedure. First, they trained the model on all the consensus peaks.
Consensus peaks, meaning what exactly?
Yeah. Basically, all the open DNA regions across the entire brain, regardless of the specific cell type.
Wait, why do that first? Why not just jump straight into the specific cells they wanted to study?
Because the AI needs to understand the general landscape of the genome first. It is like teaching someone the basic grammar and vocabulary of a language before asking them to interpret a highly technical, nuanced poem.
Oh, that makes sense. Get the baseline rules down.
Right. Once the AI understood the general rules of brain DNA, then they fine-tuned it specifically on the tiny, nuanced peaks that differentiate one specific type of brain cell from another.
And the paper notes this approach totally crushed the competition. It was significantly more accurate at predicting DNA accessibility in specific brain cells than larger alternative models like the GRLU framework.
It outperformed them handily.
But wait, here is where it gets really interesting to me. Because the model wasn't just stuck reading mouse DNA. The researchers took this AI, which had only ever seen the inside of a mouse brain, and they asked it to look at the DNA of a chicken.
Yes. They pointed the model at the chicken's UACA gene locus.
And the mouse-trained AI accurately identified the specific enhancer logic for parvalbumin, or PVALB, interneurons in the chicken. Think about that for a second. A mouse and a chicken are separated by hundreds of millions of years of evolution.
It is a massive gap.
Right. Our mammalian ancestors and the ancestors of birds diverged an unimaginably long time ago.
If you are listening right now, imagine if the passcode to your brand new smartphone somehow also perfectly booted up a dusty Windows 95 desktop computer from decades ago.
That is a great analogy.
That is the level of shared ancient history we are looking at between a mouse and a chicken. The mixing board dials, the core genetic passwords that define a specific type of brain cell, they just haven't fundamentally changed since those evolutionary lines split.
It really implies a universal biological grammar. But how granular is the AI's understanding of this grammar? I mean, is it just looking at general neighborhoods of DNA, or is it getting up close?
It achieves true nucleotide resolution, which is incredibly granular. Let me give you a specific example from the paper.
Please do.
The AI identified a specific E-box motif that is a very short sequence of DNA, and it is unique to a rare type of interneuron in the mouse called Salt-Chortle cells.
Okay.
The specific sequence is CAGGTG. The model showed that mutating just one single letter, changing that third G to a C, making it CAGCTG, it completely changes the prediction.
Just one letter.
Just one letter. That single letter flip causes the DNA to activate a completely different class of interneurons instead.
One letter out of billions in the genome. But, I mean, biologically, how does one letter actually do that physically?
Well, remember that transcription factors are physical 3D proteins. When you change that one letter in the DNA sequence, you are subtly changing the physical shape of the docking station.
Ah, so the key doesn't fit the lock anymore.
Exactly. The original protein can no longer fit, but a completely different protein suddenly fits perfectly. The AI is mapping these microscopic physical interactions purely by looking at the text of the code.
That is mind-blowing.
And it works across tissues, too. They also built a human blood cell model, the PBMC, that successfully identified known, validated transcription factor binding sites, like EBF1 in B cells and ETS1 in T cells.
Okay, time for another quick recap. Point 1, the DPICCN2 model beat the competition using a two-step training method. Point 2, it proved a shared biological grammar by reading chicken DNA using mouse rules. Point 3, it is so precise that changing a single DNA letter flips the target cell type.
Spot on. And it really makes you think. Does it surprise you that mouse and chicken brain cells share the exact same genetic passwords? It just shows how conserved this code really is.
Yeah, and if this AI is smart enough to find the exact same biological grammar in a mouse and a chicken, and it can map human blood cells with that same precision, it makes you wonder, can it find shared grammar in diseases?
That is the logical next step.
Like, can it look at different human cancers and find a common instruction manual?
That is exactly what the researchers investigated next. You see, cancer is notoriously difficult to study because of patient-specific noise. Every single patient's tumor has a highly unique epigenome, filled with random mutations and chaotic data.
So it is hard to find the signal in all that static.
Exactly. However, certain dangerous, highly aggressive cell states appear to recur across vastly different types of tumors.
Right. The paper specifically looks at mesenchymal-like, or MES, cell states. You see this state in melanoma, which is a skin cancer, but you also see it in glioblastoma, or GBM, which is a highly aggressive brain cancer.
Yes. Two completely different cancers.
But hold on, those tumors originate in completely different organs from entirely different types of tissues. How are they possibly using the same manual?
Because the cancer is essentially hijacking the genome. By using CREsted to isolate the core enhancer logic, the actual rules of the mixing board we talked about earlier, the researchers were able to filter out all that patient-specific status. And the model proved that these MES states in melanoma and glioblastoma actually share a highly similar underlying enhancer logic.
Wow.
Yeah, the tumors are located in different organs, but they are hacking into the exact same ancient genetic instruction manual to drive their aggressive behavior.
That is massive for potential therapies. I mean, if you can prove that different cancers rely on the exact same corrupted manual, you don't necessarily have to invent a unique drug for every single tumor type. You just design a drug to, burn that specific manual.
It completely changes the target of oncology. But to find that manual, the researchers had to prove their AI was actually better at this than the massive models that are dominating the tech industry right now. There's a fascinating technical showdown in this section of the paper.
Oh yeah, the David versus Goliath AI battle. The researchers compared CREsted's custom-built models against massive pre-trained foundation models like Borzoi and Informer.
And for context, these foundation models are absolute giants. Tech companies train them genome-wide on thousands of different assays. They have millions and millions of parameters.
Because the current trend in AI is basically, build it bigger, feed it more data, and it will automatically be better.
That is the prevailing assumption, yes.
Yeah.
But when they fine-tuned the massive Borzoi model to predict high-resolution, cell-type specific accessibility in the mouse cortex, the results were highly unexpected.
Let me guess. Goliath lost.
Goliath lost. The smaller model, trained from scratch via CREsted DeepICCN2, it matched or even beat the massively larger fine-tuned Borzoi model.
But why? How does a smaller model beat an AI that has seen exponentially more data?
Because giant foundation models look at the entire genome's macro patterns. They are trained to see the whole forest. But when you ask them to differentiate between two incredibly closely related brain cells, that macro view sort of blurs out the microscopic single nucleotide differences. CREsted's smaller size and targeted two-step training forces it to hyperfixate on those tiny crucial differences without getting distracted by the rest of the genome.
Yeah, that makes total sense. It is like hiring a general contractor versus hiring a master carpenter to build a highly complex custom cabinet.
I love that analogy.
Like, the general contractor knows how to build an entire skyscraper, but their tools are just too big and blunt for this. The master carpenter knows exactly how to carve the intricate millimeter-level details you need.
Very apt. It proves that in biology, context and parameter-efficient architectures often trump sheer computational volume.
Okay, quick recap time. Point 1, different cancers use the exact same enhancer logic to achieve dangerous cell states. Point 2, highly specialized, smaller AI models can actually outperform massive foundation models in these high-resolution tasks.
That is exactly right.
So to our listeners, in the current era where tech companies are just building bigger and bigger AI models, what do you make of the fact that a smaller, specialized tool won out here? It is definitely something to think about.
It challenges the whole bigger is better paradigm, which really sets the stage for what I consider the most staggering achievement in the paper. Reading the code is one thing. Filtering out the noise to find cancer states is incredible. But CREsted's fourth module goes a step further. It writes new biological code.
Yes, the AI stops observing and starts playing creator. The researchers wanted to see if they could design synthetic enhancers that actually function inside a living organism.
Right.
To do this, they built a model called Deep Zebrafish. They trained it on a massive data set of developing zebrafish embryos, covering 639 different cell type and time point combinations.
They basically gave the AI the complete encyclopedia of how a zebrafish develops from a single cell.
And then they utilized a process called in silico evolution, or IS.
Yes, in silico evolution.
Okay, here is where my mind started bending. As I understand it, they didn't start by tweaking existing zebrafish DNA. They literally started with random garbage DNA sequences generated by the computer.
Correct. Completely random strings of A, C, T, and G, sequences that do absolutely nothing in nature. And then they applied an L2 distance cost function to iteratively mutate.
No, wait, stop right there. An L2 distance cost function? That sounds like a grad school math exam. What are they actually doing to this garbage DNA? I need a translation here.
Fair enough. Think of the cost function as a penalty score. The AI looks at the garbage DNA and asks its predictive model, if I put this in a fish, what will happen? The model says, nothing. That results in a really high penalty score.
Okay, so high score is bad.
Exactly. So the AI randomly mutates a few letters, changes an A to a C, a T to a G, and it asks again. It runs thousands of simulated mutations, constantly keeping the changes that lower the penalty score.
Oh, I see.
The goal is to evolve the sequence until the model predicts it will activate only in one specific target, like the cardiac muscle, and will remain completely silent everywhere else in the fish's body.
It is like writing a highly customized, encrypted delivery address on a biological package. The AI is refining the text over and over in a simulation so that only the heart cells possess the exact physical key required to open the package, while the rest of the body just ignores it entirely.
And this is where the paper delivers its ultimate climax. Having a computer predict that a synthetic DNA sequence will work in a simulation is neat, right? But they actually synthesized these computer-generated sequences in a physical lab and injected them into living, breathing zebrafish embryos.
And the fish could read it.
The living organism recognized it as valid functional code. The synthetic enhancers specifically lit up the cardiac muscles and the somatic muscles, exactly as the AI predicted.
That is wild.
The computer wrote a brand new piece of DNA from scratch, and it worked in vivo.
But wait, hold on. Biology is usually highly specialized. Doesn't that risk the AI crossing its wires? Like, what happens if you want to target more than one thing? The paper mentions dual specificity enhancers.
Yes, to really flex the model's capabilities, they tasked the AI with creating a single piece of DNA program to activate in both cardiac and somatic muscles simultaneously, but at specific mathematical ratios.
So not just on or off, but precise volume control for two different organs at the same time.
Exactly. They asked for sequences that would drive expression in a 1-to-1 ratio, or a 2-to-1 ratio between the two distinct muscle types.
Writing dual code logic. But does that actually work? Doesn't the DNA just get confused and misfire?
Well, it worked. The expression efficiency, basically how powerfully the genes turned on overall, was a bit lower for these dual target enhancers compared to the single target ones, which isn't entirely surprising given the complexity.
Right. It is doing double duty.
But the sequences successfully converged. They achieved the precise dual targeting ratios. And when the Huey researchers analyzed how the AI solved this puzzle, they found the computer had independently utilized known biological motifs like GATA, NKX, and TED.
So it wove those together to build this synthetic code.
Yes, the AI independently derived the ancient rules of biology to engineer a completely novel, customized solution. The blueprint isn't just a read-only document anymore. It is fully programmable.
Okay, final recap for today. Point 1, the deep zebrafish model evolved random garbage DNA into working synthetic enhancers. Point 2, these computer-generated sequences successfully targeted specific organs in living fish. Point 3, they even created dual-target DNA with specific expression ratios.
A huge leap forward. And if we connect this to the bigger picture, we're moving from observing biology to programming it. For everyone listening, how does that shift change how you view the future of medicine?
We've gone on quite a journey today. I mean, we started with the abstract concept of an enhancer code, the degenerate mixing board of cell identity. We watched the specialized AI read that code across hundreds of millions of years of evolution, proving that mice and chickens share the exact same genetic passwords.
Truly incredible.
We saw it cut through the chaotic noise of human cancer to find shared vulnerabilities where massive foundation models actually failed. And finally, we watched it write brand new synthetic DNA that functions perfectly inside a living organism.
It represents a staggering leap in our capability to interface with life itself.
And that leads to a final, really provocative thought for you to mull over.
Yes. The researchers proved they can write a dual code enhancer that targets two specific cell types at once in a zebrafish. Imagine applying this exact same logic to human therapies. Consider CAR T-cell therapies, where we engineer a patient's own immune cells to hunt down and attack cancer.
Right.
Right now, a major, sometimes fatal challenge is that these therapies experience collateral damage. They attack healthy tissue that happens to look slightly like the tumor.
Because the targeting isn't specific enough, it is essentially a blunt instrument.
But what if we could design synthetic DNA for those CAR T cells that requires a dual code to activate? We could program the immune cells with complex IFIN logic. The synthetic enhancer would only activate the immune attack if it detects marker A and marker B simultaneously on the target cell.
Oh, wow.
If it bumps into healthy tissue that only has marker A, the enhancer remains silent, leaving the healthy tissue completely untouched.
We're talking about writing complex, multi-conditional software logic directly into the human genome. It is no longer science fiction, it is just a matter of time. Thank you so much for joining us on this deep dive. Keep questioning the blueprints around you, and we will see you next time.